Hello friends, welcome to PVIC Breakdowns. In this video, I will be discussing the metabolism of lipids. Particularly, I will be discussing the breakdown of dietary or ingested lipids. That is lipids gotten from the food we eat. Now, what is lipid metabolism? Lipid metabolism simply involves the synthesis and breakdown of lipids in the body. Now, most of the lipids found in the food we eat and present in our body are triglycerides, cholesterol esters, and phospholipids. Now, this whole process involving the breakdown of dietary lipids can be divided into 3 stages. The first stage being lipid digestion. Now, in this first stage, the triglycerides present in the ingested food are broken down into smaller molecules such as diglycerides and free fatty acids by the enzyme lingual lipase. And this enzyme lingual lipase is produced and secreted in the tongue. So let's see how lingual lipase works. Let's see, this is the lingual lipase enzyme, and this is the triglyceride molecule. So we have the glycerol backbone, and we have the 3 fatty acid chains, the triglyceride molecule. So lingual lipase hydrolyzes a fatty acid chain from this triglyceride molecule to produce a diglyceride and a free fatty acid. So this is how lingual lipase works. However, these other lipids, cholesterol esters, and phospholipids, are not acted upon by this enzyme or any enzyme until they reach the small intestine. Now, from the mouth, the digestion of triglycerides continues into the stomach. In the stomach, the enzyme called gastric lipase. This enzyme continues the digestion of triglycerides to produce diglycerides and free fatty acids as well. So these 2 enzymes, lingual lipase and gastric lipase, only carry out the partial digestion of triglycerides. Most of the digestion of triglycerides and lipids in general occurs in the small intestine. Now, the lipid contents from the stomach, now moves into the small intestine, and most of these lipids are still undigested and are in the form of large lipid droplets. So a hormone called cholecystokinin, this hormone stimulates the release of bile from the gallbladder. Now, bile is a substance that is produced by the liver and stored in the gallbladder. So this hormone now stimulates the release of bile from the gallbladder into the small intestine. And bile consists of mostly water, bile salts, bilirubin, and even cholesterol. So the bile salt component of bile helps in the emulsification and or breakdown of lipids. So let's see how this bile salt works. Let's say this is the large lipid droplets. And this is the bile salts. Bile salts have a hydrophobic side and the hydrophilic side. It is this hydrophobic and hydrophilic property of bile salts that enables them to break down large lipid droplets into smaller droplets. So at the end we have something like this. These are smaller lipid droplets. So these are smaller droplets. And this breakdown increases the surface area of lipids and makes them more accessible to digestive enzymes. And also remember, our basic chemistry tells us that an increase in the surface area increases the rate of a reaction. So this is the whole emulsification process. And at the same time, this same hormone cholecystokinin stimulates the pancreas to release a substance called pancreatic juice. Now, pancreatic juice consists of so many enzymes, amongst which are pancreatic lipids, colipids, cholesterol esteres, and phospholipids A2. Now, pancreatic lipids carries out most of the digestion of triglycerides to produce monoglycerides, free fatty acids, and glycerol. Colipids has no catalytic activity. It only helps to increase the efficiency of pancreatic lipids. Cholesterol esteres helps to break down some of the cholesterol esters to produce free fatty acids and cholesterol. Phospholipids A2 helps to break down some of the phospholipids to release free fatty acids. Now let's see how these two enzymes work. Cholesterol esteres and phospholipids A2. Let's see how they work. Now for cholesterol esteres, let's say this is a cholesterol molecule. So you have these points. You have an OH group here. So this is a cholesterol molecule. Now for a cholesterol ester is simply a cholesterol molecule with a fatty acid chain attached to it at this point. So a cholesterol ester will look like this. This is the fatty acid chain. So this is the cholesterol ester. You can see the fatty acid component and the cholesterol component of the cholesterol ester. So cholesterol esteres, we hydrolyze this bond to release a free fatty acid and a cholesterol molecule. Now for phospholipids A2, let's draw a phospholipid. So this is the phospholipid. These are the two fatty acid chains attached at position 1 and position 2 of the phospholipid, and this is a phosphate group with its head group attached at position 3. So phospholipase A2 will cleave this fatty acid chain from position 2 of the phospholipid. So phospholipase A2 will hydrolyze this bond to release a free fatty acid. Now, this is how cholesterol esterase acts on cholesterol esters, and this is how phospholipase A2 acts on phospholipids. Before we move further, let's take note of something. Cholesterol and phospholipids are both amphipathic molecules. Amphipathic. This means that they have hydrophilic and hydrophobic properties. Now, for a cholesterol molecule, this is the hydrophilic part. This OH group is the hydrophilic part, and this remaining portion is the hydrophobic part of the cholesterol molecule. Now, for a phospholipid, these two fatty acid chains make up the hydrophobic part, while this glycerol backbone and the phosphate group with its head group make up the hydrophilic parts. So this is why cholesterol and phospholipids are amphipathic molecules. And you have something like this for phospholipids. We use this to represent phospholipids. So this is the hydrophilic head consisting of the glycerol backbone and the phosphate group. And these are the hydrophobic tails that represent the fatty acid chains. Now, let's move further. And another thing we should take note of is the digestion by enzymes involves mostly the triglycerides. The digestion by enzymes involves mostly the triglycerides. So let's move to the next stage, which is lipid absorption and transport. Now, the product of lipid digestion, such as the free fatty acids, glycerol, cholesterol esters, and so on, this product of lipid digestion need to get into the bloodstream. But how? Now, in this process, the product, let's say these are the products of lipid digestion. The products of lipid digestion are now surrounded by bile salts. These are bile salts surrounding the product of lipid digestion to form structures called micelles. Micelles. So these micelles consist of a lipid core consisting of the products of lipid digestion and bile salts clustering around. Now, so these micelles now enables the efficient transportation of lipids to the epithelial cells of the intestinal villi. That brings us to this diagram. So now this is a small portion of the small intestine. Now this is how the small intestine looks like. And if we enlarge these parts, if we just enlarge these parts, so this is what it will look like. And these many tiny projections are called villi, plural villi, singular villus. So if we now project a villi, we decide to enlarge a villus. If we enlarge a villus now, this is what it looks like. You can see the arrangement of the epithelial cells. These are the epithelial cells. Then you have the capillary network at the middle and a lymphatic vessel called lacteal enclosed within. So this is the arrangement. So when these micelles now, when they reach the intestinal villi, the product of lipid digestion dissociates from the micelles, thereby allowing the bile salts to continue their work, while the product of lipid digestion moves into the intestinal epithelial cells. Now, when inside the intestinal epithelial cells, some of the products of lipid digestion, such as short chain fatty acid, medium chain fatty acids, and glycerol, because of their small size, diffuse directly into the bloodstream through the capillary network. However, larger lipids such as the long chain fatty acids, cholesterol esters, phospholipids, and so on do not diffuse directly into the bloodstream, but rather are packaged into transport particles called chylomicrons. And note that the long chain fatty acids and monoglycerides are reassembled back into triglycerides. So this chylomicron is a lipoprotein particle. That consists of an inner core of triglycerides, you can see the triglycerides, and cholesterol esters, and an outer membrane of phospholipids. These are the phospholipids mixed with cholesterol molecules and a protein called apolipoprotein. So this is the chylomicron particle. And the amphipathic nature of phospholipids and cholesterol, together with the binding of this apolipoprotein, makes chylomicrons water soluble and transportable through the watery environment of the bloodstream. So a chylomicron is a lipoprotein particle that serves the primary purpose of transporting dietary lipids from the small intestine to body tissues that need them, such as the skeletal muscle, the cardiac muscle, and adipose tissues. Now, when this product of lipid digestion have been packaged into chylomicrons, these chylomicrons then leave the epithelial cells. They leave the epithelial cells and move into this lymphatic vessel called lacteal. So this lacteal consists of lymph, just like capillaries consist of blood. So just like the blood in capillaries, the lymph in this lacteal transports the chylomicrons through the lymphatic network to the largest lymphatic vessel called the thoracic duct. It is at this point that the chylomicrons enter the bloodstream. Now that takes us to the next stage and the final stage, which is lipid catabolism. Now, in lipid catabolism, this is the diagram for lipid catabolism. Now, for lipid, the chylomicrons are transported through the bloodstream. So, when they reach their destination, such as the skeletal muscle cell, they are now broken down by an enzyme called lipoprotein lipase, and this enzyme is present on the surface of endothelial cells. Now, what are endothelial cells? Endothelial cells are cells that line or cover the surface, like lining of jackets, the inner lining of jackets. So these endothelial cells line or cover the inner surface of blood vessels, just like epithelial cells cover the surface of the small intestine. So this lipoprotein lipase is found on the surface of endothelial cells, and it breaks down these chylomicrons to release triglycerides. Now, these triglycerides are still broken down by the same enzyme to release free fatty acids and glycerol. These free fatty acids and glycerol then diffuse into this space, which consists of interstitial fluid. Now, interstitial fluid is a body fluid that is found between capillaries and that is found between blood vessels and cells. And this interstitial fluid consists of nutrient molecules that have diffused from the capillaries, and it also consists of waste product that has been discharged from the cell. So the free fatty acids and glycerol in the interstitial fluid are now transported into the cell. Now when inside the cell, the glycerol component, the glycerol is converted to glyceraldehyde 3-phosphate, which can now enter into the glycolytic pathway to provide energy in the form of ATP. Now as for the free fatty acid, the free fatty acid is then transported into the mitochondria of the cell. Now in the mitochondria, the free fatty acid undergoes beta oxidation. And basically, the beta oxidation of fatty acids produces acetyl-CoA. This acetyl-CoA then enters the TCA cycle, and subsequent reactions now leads to the production of energy in the form of ATP. So this is about lipid catabolism. You can also watch videos on beta oxidation of lipids to have a clear understanding on beta oxidation. So this is all about lipid metabolism of dietary lipids. I hope you enjoyed this lesson. Kindly share with your friends, and don't forget to click the like button and subscribe to my YouTube channel. Thank you and God bless you.